Can you see my slide?
Not yet. Yeah, I can see
Okay.
now.
Oh, okay, fine. So layers of retina. Actually, the light rays, it travels in this direction. Yeah, it comes in this way. That is, the light rays comes from here. It passes the cornea, then pass the iris, then through the pupil, then lens, and finally falls on the retina. And actually, the photoreceptors, when we view it in this manner, in this like in this picture, the photoreceptors or the rods and cones, they are seen towards the outermost part of the retina. Okay, they are seen towards the outermost
Hmm.
part of the retina. So what happens is those light rays, they'll penetrate through this area, they'll enter like this inside, and after entering, they'll pass through this layers, that is through all these layers. This is the retinal part, and it will pass through all these layers and stimulate the photoreceptors. Okay,
Yeah.
that's how it happens. So the light rays, it's just in an inverted manner. It's not like the photoreceptors are located in this part of the retina, that is inside the part or the area where light falls. It's not that the photoreceptors are located there. They are just in the outer part of the retina. It is seen very close to the choroid layer, that is the second layer or the vascular tunic of eye. Okay, so light rays, they'll pass or they'll penetrate through all these layers and reach the outermost area, and from there these photoreceptors will accept those light rays, convert them into electrical impulses or electrical signals, and then it will move in this direction. I hope you got the point.
Hmm.
Any doubt?
No.
Okay. So actually there are 10 layers of retina. Okay, it is divided into 10 layers, and it is starting from the front portion. Okay, wait, starting from this part. Yes, starting from this part, this area, this section of the retina. I'm coming from outer towards the inner part. Okay,
The
outer
light
towards.
rays fall on the inner part, no?
Yes, light falls on the inner part. That's what I said previously, that
Hmm.
light will fall in the inner part. It will fall here. Okay. So what happens is that light rays, it will penetrate through all these layers, because these layers cannot convert them. The light rays, they cannot convert those light rays into electrical simple impulses. So the light rays, they'll penetrate through this entire nerve cells and reach
Hmm.
the outermost part, then stimulate the rods and cones.
Hmm.
They'll stimulate the rods and cones, or the light rays will fall on this rods and cones, and it is then these rods and cones convert those light energy or light rays into electrical impulses, and then those electrical impulses will be transmitted to these various layers, and it will reach back into the inner part of the retina. You got the
Hmm.
point?
Yes.
Okay. Okay, so there are 10 layers, and I'm going to start with the outermost part, with this part. Okay, I'm coming from here to the inner part of the retina. So the first layer is called the pigmented epithelium. It is the outermost layer, and it is seen very adjacent to the choroid. Okay, it is the outermost layer of retina, which is seen very close to the choroid layer.
Hmm.
Then it contains numerous pigmented granules, like round pigmented granules, and all those extensions, they'll pass on to the rods and cones, or these granules, they have their extensions in between the rods and cones. Okay, and it also contains those melanin colored melanin pigments called fusin, and it is in this pigmented epithelium vitamin A is stored. Okay, vitamin A is stored in this pigmented epithelium of the retina. So it is the outermost layer seen very close to the choroid layer. It contains numerous pigmented granules, and it is in this layer vitamin A is stored. Okay, and any damage to this pigmented layer or pigmented epithelium may lead to deficiency of vitamin A, which in turn leads to the disorder or the problem called night blindness. Okay. Then next comes the photoreceptor layer. That is just after the pigmented epithelium, we can see the photoreceptor layer that contains the rods and cones, the visual receptors. So the light rays will fall on these rods and cones, and it is these photoreceptors that convert those light energy or light rays into electrical impulses. Okay. Then next layer is the external limiting membrane. Okay, it is a very thin layer, and it contains numerous supporting fibers or supporting elements called Muller fibers. Okay, it is a very thin layer seen in between the photoreceptor layer and the next layer. It is a very thin layer made up of many fibers called Muller fibers. Then comes the outer nuclear layer, which is formed by fibers and granules of rods and cones. So the extensions or the end part, the axon part or the tip portion of rods and cones are seen in the outer nuclear layer. Then comes the outer plexiform layer. It also contains the terminal fibers of rods and cones and dendrites from bipolar cells. So in this layer, that is in the outer plexiform layer, we can see those connections or those synapses between the terminal fibers of rods and cones and the dendrites of bipolar cells or the bipolar neurons. Then next comes the inner nuclear layer. The previous one was outer plexiform layer. Then comes inner nuclear layer. It contains the bipolar neurons or the bipolar cells. So these bipolar cells, they have got connections, or they'll pass the information. They'll collect the information from rods and cones, and they'll pass the information to another group of cells or neurons called ganglionic cells, which is seen in the next portion. So this bipolar cells or bipolar neurons, they have or they form synapses with the photoreceptors as well as ganglionic cells. So the this one dendrites of bipolar neurons or the bipolar cells will form synapse, or they'll form synapse with the photoreceptors, the terminal ends of rods and cones, and the axons of this bipolar neurons will form synapse with dendrites of ganglionic cells. So the main function of bipolar cells is to collect the electrical impulses from photoreceptors and transfer them into the ganglionic cells. Is it clear? Okay. Then next comes, and along with these structures, there are also some association neurons, that is interneurons or association neurons. They help in transferring the information along with the bipolar cells. Okay, they are connecting neurons. We can term them in many ways: interneurons, connection neurons, or association neurons. So this inner nuclear layer also contains association neurons called horizontal cells and amacrine cells. They are all association neurons. They are not coming in direct. This one, what they do not play a direct role in the transferring of information, but they help in supporting the bipolar neurons. Okay, and in some cases they'll also help in formation of synapses with the ganglionic neurons or the photoreceptors. Okay, that's the function of horizontal cells and amacrine cells. Then next is inner plexiform layer, which consists of synapses between dendrites of ganglionic cells and axons of bipolar cells. So it's just a layer in which we can see the synapses only, just the synapses, the synaptic transmission, or just the junction between the ganglionic cells and axons of bipolar neurons. Then next comes the ganglionic cell layer. ganglionic cell layer, they contain multipolar neurons. And they have lots of multipolar neurons, and the axons are seen in inner surface of retina, and they form the optic nerve. So it is actually the axons of ganglionic cells that form the optic nerve. Okay, it is the axons of the ganglionic cells that form the optic nerve. And then comes the nerve fiber layer formed by non-myelinated axons of ganglionic cells. Now, from this layer onwards, that is from ganglionic cell layer onwards, it is actually the part or the innermost part of the retina. Okay, these layers form the innermost part of the retina. We are coming from outer part towards the inner side. So ganglionic cell layer and the nerve fiber layer form the innermost part of the retina, and nerve fiber layer sorry, it is formed by non-myelinated axons of ganglionic cells. And last comes the internal limiting membrane, which is the innermost layer that separates retina from the vitreous body. That is, it separates retina from the vitreous humor or the vitreous area. Okay, so these are the layers of retina. You want me to repeat it once again?
The inner nuclear layer, inner plexiform layer.
Yeah. No. First one, before that. Yeah. The first one is pigmented epithelium. When we come from the outer side, the pigmented epithelium. Then comes the photoreceptor layer that contains the rods and cones. Then comes the external limiting membrane. Then outer nuclear layer formed by fibers and granules of rods and cones. The external limiting membrane, then outer nuclear layer, and the outer plexiform layer. These three layers contains the photoreceptor parts. Okay, the various areas or various parts of photoreceptors, rods and cones, their axon part, then their axonite part, etc. So these three layers have those areas of rods and cones. Then after that, after outer plexiform layer, we can see the inner nuclear layer where the bipolar neurons are seen. Okay, it is in the inner nuclear layer the bipolar neurons are seen, and these bipolar neurons along with horizontal cells and amacrine cells form the inner nuclear layer. Okay. Then next comes inner plexiform layer. It consists of synapses between dendrites of ganglionic cells and axons of bipolar cells. So this layer contains the synapses. It's just their connection only. There are no cells or there are no neurons present in that particular area. We can see just the synapses between them. Okay. Then comes ganglionic cell layer, which is another group of neurons seen in this, another group of neurons, the ganglionic neurons or ganglionic cells. And it is the axons of the ganglionic cells or the ganglionic neurons that form the optic nerve. They are seen in the inner surface of retina and they form the optic nerve. And this ganglionic cell layer and nerve fiber layer together form the innermost part of the retina. It forms the innermost part of the retina. We can say it as the sensory retina. Okay, it is these layers, that is the inner layer, inner nuclear layer, inner plexiform layer, ganglionic cell layer, and nerve fiber layer that form the areas of sensory retina. And the previous ones, outer plexiform layer, then outer nuclear layer, then photoreceptor layer, and the pigmented epithelium, all of these form the pigmented retina. Is it clear? Okay. And the last one is the internal limiting membrane, which is the innermost layer that separates retina from vitreous body. So the vitreous humor and the innermost layer or the nerve fiber layer of retina is separated by this internal limiting membrane. Okay, so these are the layers. Now here it is. So we'll start with this one. This is the choroid part. Okay, this is from here we'll move towards the retinal portion. So this is the outer part of the retina. First part is the choroid layer. After choroid layer, we can see the pigmented epithelium. Okay, pigmented epithelium, the outermost layer of retina. Then after that, after the pigmented epithelium, it is here in this pigmented epithelium, vitamin A is stored. Then after the pigmented epithelial layer, we can see the photoreceptor layer. So this photoreceptor layer, this part, photoreceptor layer contains the outer part or the upper part of the photoreceptors, rods and cones. This conical cells are cones, and this one flattened ones are the rods. Okay, so this is the photoreceptor layer. Then comes the outer limiting membrane or the external limiting membrane, this small line, this part. This is the only part, a very thin layer. Okay, that is the external limiting membrane. They are the chief supporting elements. Okay, then after that comes the outer nuclear layer. So this outer nuclear layer, you can see that the outer nuclear layer contains the nuclei or the granules of rods and cones. See, these are the granules, the rounded parts. So this outer nuclear layer contains the granules of rods and cones. Then comes the outer plexiform layer. So in this part, in this outer plexiform layer, we can see their synapses. See, this is from the photoreceptors, and these are the dendrites of the bipolar neurons. Okay, so here we can see the synapses between each this one, the rods and cones or the photoreceptors and the bipolar cells. So this is the outer plexiform, sorry, inner nuclear layer. No, we were saying about outer plexiform, sorry. Outer plexiform layer. So it contains the synapses between bipolar neurons and the photoreceptors. Then comes the inner nuclear layer, where we can see the bipolar neurons very clearly. Okay, we can see the bipolar neurons very clearly. The body part is seen here, and along with bipolar neurons, we can see hemacrine cells as well as horizontal cells. Okay, hemacrine and horizontal cells are seen. That is the inner nuclear layer. The next one is the inner plexiform layer, in which we can see the synapses between bipolar neurons and the dendrites of ganglionic cells. Inner plexiform layer. We can see the synapses between bipolar neurons, the axons of bipolar neurons and the dendrites of ganglionic cells. Then next comes the ganglionic cell layer, where we can see the ganglionic neurons or the ganglionic cells. And after that comes the nerve fiber layer, where we can see the end portions of these ganglionic neurons, and they finally form, or this nerves combine together to form the optic nerve. And the last one is the inner limiting membrane, which is a very thin membrane, just like external limiting membrane. Very thin membrane that helps in separating these layers of retina from the vitreous humor. Is it clear now? Okay. Once again, the same picture has been shown here also. So choroid, after that comes the pigmented epithelium. Then we can see the photoreceptor layer. Then the external limiting membrane, which is a very thin layer, this one. Then comes the outer nuclear layer, where we can see the granules of the rods and cones. Then comes outer plexiform layer, where we can see the synapses between photoreceptors and the dendrites of bipolar neurons. Then comes the inner nuclear layer, where we can see the bipolar neurons as well as the horizontal and hemacrine cells that help in supporting these bipolar neurons. After that comes the inner plexiform layer, where we can see the synapses between bipolar neurons and the ganglionic cells or the ganglionic neurons. Then comes ganglion cell layer, where we can see the ganglionic cells or the ganglionic neurons. And finally comes the nerve fiber layer, retinal nerve fiber layer, where the axons of this ganglionic layer or the ganglionic cells combine together to form the optic nerve.
Mhm.
And the last one is internal limiting membrane, which separates these layers of retina from the vitreous humor. Clear now?
Yeah.
Okay. And this is how the light rays pass. I told you in the beginning. Okay. So light comes in this.
Wait, what is the purpose of amacrine and horizontal cells?
They are supporting cells with bipolar cells. They are supporting. They help in transferring the information. Some of the information from these neurons or from the photoreceptors. They help in accepting some of the information from these photoreceptors. They are just supporting neurons.
Mm.
They support the bipolar neurons. That's all.
Okay.
Okay.
Hmm.
So light rays, this is the picture. So light rays will fall on the inner part. That is, it will actually fall on the inner layer of inner layer of the retina, the inner limiting membrane of the retina. But since photoreceptors are located in the outer part of the retina, that is in the pigmented section, these light rays will penetrate through all these layers. But these light rays will not be accepted by these neurons because they have no information, or they are not the structures, or they are not the receptors for converting these light rays into electrical impulses. So the light rays, they'll pass through all these layers and finally reach this photoreceptor layer. And it's these it's these rods and cones that accept these light rays, and these flattened portion of rods and the conical portion of the cones, they actually act as the receptor portion or the receptor sites. So the light rays will stimulate these areas, and these areas will pass the information to this granule area of the photoreceptors. And it is this granule area that converts these light energy or light rays into electrical impulses, and through this axon terminals, the information or the electrical impulses will be transmitted to bipolar cells. Okay, this is how it happens. Okay, the same thing over here also. So that much about the layers of retina. Is it clear? Okay. Any doubt?
No.
Oh, okay. And next, we'll move on to countercurrent mechanism. And before speaking about countercurrent mechanism, we have to first know how dilute and concentrated urine are formed in the kidneys. Okay, that is the first part. Only after understanding that section, you will be able to understand this countercurrent mechanism. And for that, firstly, we have to know the speciality of various parts of, speciality of cells lining the nephron. Okay. About the nephron, firstly, the structure and everything, it's like this. And we have the nephron is actually divided into two major parts, as the renal corpuscle and renal tubule portion. Renal corpuscle contains the glomerulus and Bowman's capsule, and renal tubule portion contains proximal convoluted tubule or PCT, then loop of Henle, then distal convoluted tubule, and finally the collecting tubule. So these are the parts of renal tubule. And loop of Henle is again divided into three, as the descending limb of loop of Henle, the hairpin turn, and the ascending limb. So these are the parts of nephron. This is the glomerulus, that is a tuft of blood capillaries or a network of blood capillaries. And it is of arterial in nature. That is arterial blood, or the oxygenated blood will pass through these blood vessels. Okay. And then comes the Bowman's capsule. Bowman's capsule is followed by the proximal convoluted tubule. Then comes the descending limb of loop of Henle. This is the hairpin turn, the U-shaped turn. Then comes the ascending limb of loop of Henle, then DCT, distal convoluted tubule, and finally the collecting tubule or the collecting duct. So these are the parts of nephron. Then to say about the peculiarities of the cells lining these parts, that especially the tubule portion of nephron. Okay, so the peculiarities are, firstly, in the case of proximal convoluted tubule, the cells in proximal convoluted tubule, they are equally permeable to both water as well as solutes. Okay, they don't have any difference or they don't have any partiality. The proximal convoluted tubule or the cells in proximal convoluted tubule are equally permeable to both water as well as solutes. Now, when we come to the descending limb of loop of Henle, it is in this picture, it is shown as a very thin part. Okay, so that indicates that the descending limb of loop of Henle is highly permeable to water and negligibly or almost impermeable to solutes. Okay, it is highly permeable to water and impermeable to solutes. Then this hairpin turn of loop of Henle, the U-shaped tube of loop of Henle, then the ascending limb of loop of Henle, and even the DCT. These three areas, that is the hairpin turn of loop of Henle, the ascending limb of loop of Henle, and DCT, all these parts are highly permeable to solutes and impermeable to water. Did you get the point?
Yeah.
Okay. And lastly, collecting tubule or collecting duct. It is normally impermeable to water. Okay, it is normally impermeable to water. But when we have or when our body experiences dehydration or decreased water content, at that time, this collecting tubule will become permeable to water by the action of a hormone called ADH, anti-diuretic hormone. Have you heard of that hormone?
Yes, yes.
Okay. So by the action or with the help of ADH, collecting tubule will become permeable to water by inserting certain water permeable carrier proteins called aquaporins. I hope you know that also.
Yeah.
Okay. So that is about the peculiarities of cells lining the tubules. This one. Peculiarities of cells lining the renal tubules. Firstly, PCT, it is equally permeable to both water and solutes. Then descending limb, it is highly permeable to water and negligibly permeable to solutes. Then comes loop of Henle, ascending limb and DCT. They are highly permeable to solutes and impermeable to water. And finally, collecting tubule, which is normally impermeable to water but becomes permeable to water during dehydration by the action of ADH.